Hello all, I am Brinda from the Department of Biology, St. Mary's College, Thrissur. Here I am going to present about cell motility. Cell motility is the spontaneous movement of a cell from one location to another by consumption of energy. Cell motility is required for a number of activities to occur within the body. Cell motility are of mainly two types: amoeboid, ciliary or flagellar movement. Let's go through flagella movement. Flagella. Bacterial flagella are long and thin appendages free at one end, and they are the organs for locomotion and are so thin that a single flagella can only be observed with a light microscope after staining with special flagella stains. A bacteria flagella is not a straight but helical and consisting of three distinct parts: filament, hook, and basal body. Filament is thinner, elongated terminal part attached to the hook, and it is fine, composed of fibrin protein called flagellin. A cross-section of the filament reveals that there are 8 flagellin molecules surrounding a hollow cylinder. Hook is broader and thinner unicellular basal region of a flagellum and passes out through the cell wall. It is made up of a single protein and functions to connect the filament to the basal body of the flagellum. Basal body, the major portion of the flagellum. Basal body is the major portion of the flagellum. Basal body is the motor portion of the flagellum, and it is most complex part of the flagellum. In gram-negative bacteria, the basal body have 5 rings. These are L, P, S, M and C. These rings are connected to the central rod. Then this is the typical picture of flagella. And the outer L and P rings remain embedded in the lipopolysaccharide and the peptidoglycan layers. The inner S and M rings are located with the plasma membrane, and the innermost C ring is located in the cytoplasm. In gram-positive bacteria, which lacks the outer lipopolysaccharide layer, S, M and C rings, there are a pair of protein called MOT A and MOT B that surrounds the inner ring and are associated to the plasma membrane. In addition to the MOT proteins, there is a final set of another proteins called Fili proteins. That is, Fili G, Fili M, Fili N are the most important Fili proteins. Different types of Fili proteins that are associated in the flagella. It is associated with C ring located in the cytoplasm, and the portion of the basal body that rotation is consisted of the rod, the M ring and the MOT and Fili proteins. The MOT proteins drives the motor, causing a torque that rotates the filament, whereas the Fili protein function as the motor switch, reversing rotation to signal response to the signals sent by the bacterial cells. The pattern of the flagellar arrangement is a good identification mark in bacteria. Flagella either confined to the pole or poles, or it may be present all around the body of the bacteria. Bacteria can be grouped together as under on the basis of flagellation: Atritrichus, Monotrichus, Amphitrichus, Cephalotrichus, Petritrichus, Lophotrichus. Atritrichus, that is the bacteria that lack flagella. Monotrichus, that is single flagellum on either of the poles of the bacterial cells. Amphitrichus, one flagellum or more on each pole of the bacterial cell. Lophotrichus, flagella in groups present on one pole of the bacteria. Petritrichus, then the bacteria that have the flagella present all around the body of the bacterial cells. And it's all about flagella. Then we go through the next section, cilia. The cilium is a plural. Cilia is a membrane-bound organelle found on most types of eukaryotes and certain microorganisms known as ciliates. The cilium has a shape of a slender thread-like projection that extends from the cell surface of the much larger cell body. The multicellular organisms cilia move fluid and particulate material through various tracts. In humans, ciliated epithelial lining the respiratory tract propel mucus and trap debris away from the lungs. There are two types of cilia, two classes of cilia, motile and non-motile cilia, each with a subtype giving four types in all. And this is the picture of cilia. The structure of the cilium core is called axoneme and it contains an array of microtubules. Most motile cilia have a central pair of single microtubules surrounded by 9 pairs of double microtubules called 9 plus 2 array, and the non-motile cilia have a 9 plus 0 axoneme that lacks central pair of microtubules. The cilium is assembled and built from the basal body on the cell surface. From the basal body, ciliary rootlets are emerging. Axoneme. Inside a cilium, the microtubule-based cytoskeletal core called the axoneme, and the axoneme of a primary cilium typically has a ring of 9 outer microtubule doublets. That is called 9 plus 0, and the axoneme of a motile cilium has, in addition to the 9 outer doublets, 2 central microtubule singlets are there, called 9 plus 2 axoneme. And the length of axoneme vary from few microns to 2 millimeter. And the axoneme in a motile cilium provides tracks for the microtubule motor proteins of kinesin and dynein. And the ciliary components is carried out by intraflagellar transport, which is similar to the axonal transport in a nerve fiber. Next, the basal body of the cilium. It is the foundation of the cilium is the basal body. Foundation of the cilium is the basal body and is termed applied to the mother centriole when it is associated with the cilium, also known as kinetosomes, basal granules, proximal centriole. It is a hollow cylindrical body, and in protoplasm there are 9 groups of tubules. Each tubule is formed of 3 units. 2 units extend into flagellum and third one ends between basal body and flagellum. Ciliary rootlet. The ciliary rootlet is a cytoskeleton-like structure that originates from the basal body, extends to cytoplasm, and it helps to anchor basal body with the cilium. The transport is bidirectional, and cytoskeletal motor proteins, kinesin and dynein transport ciliary components along the microtubule tracts. Functions of flagella and cilia. Helps in locomotion in flagellate and ciliated organism. Create current for obtaining food from aquatic media. And in some protists and animals like organelles, take part in capturing food. The canal system of porifers operates with the help of flagella. Internal transport of several organs is performed by cilia. Example, passage of eggs in oviduct, passage of excretory substances in the kidney, etc. And in aquatic organisms, cilia create currents in water for the renewal of oxygen supply and quick diffusion of carbon dioxide. Sometimes cilia and flagella show sensitivity to light, temperature, etc. Thank you.